Hi. Thank you very much for the organizers to give me the opportunity to present my work, which this is my postdoc work, which I did in Yasha Yeshu lab in the Harvard University. And today I would like to talk with you about goosebumps and how your stem cells can actually feel the weather. Somatic stem cells are a major player in maintaining homeostasis. Hence, in the last 60 years, many efforts have been done in order to try to understand what controls their behavior, focusing mainly on their immediate niche and these locally secreted factors. For my postdoc studies, I wanted to ask a bit different questions, which is how the environment can be integrated into stem cell function and how these changes can be felt and allow the organism to adapt to the constantly changing environment. For this, I use the skin as a model as it's in constant contact with the outside world. And specifically, I use hair follicle stem cells, which are the major cellular force that drive hair growth. So talking about changing in the environment, and a very good example is the weather. And at least in Boston, we all know that winter is coming and it's getting very, very cold. So your first immediate reaction when you are exposed to the cold is goosebumps. Goosebumps is the reaction of the contraction of the small smooth muscle, which connected directly to the hair follicle stem cells. It pulls the hair to stand erect, hence trapping a very thin layer of air and facilitating thermoregulation. This reaction is mediated by the sympathetic nerve, which is activated in a fight or flight mode, and the secretion of norepinephrine. And if you ever wonder whether mice can get goosebumps, they do. This is a control mice held in a thermoneutral environment, and you can see all the fur is lying flat. But when this mouse is exposed to the cold, you can see that he gets goosebumps and all the hair stands erect. So goosebumps provides very fast and immediate relief from the cold. But what happens if the cold persists longer and there is a requirement for additional layer of adaptation? To ask that whether the skin and specifically hair follicle stem cells can adapt to a different environment, we put mice into cold for 2 weeks and compare between thermoneutral grown mice to cold exposure. So mice that were exposed to the cold, not only that they got goosebumps, they also got specific elevation of norepinephrine content in the skin, but they also grew hair, which means their hair follicle stem cells were activated, allowing them additional layer of adaptation against the cold. Very similar results we saw when we just apply adrenergic agonist on top of the skin. All of these suggest that hair follicle stem cells can adapt and can sense the difference in the environment, and this adaptation is mediated by the sympathetic nerve. But these are extreme cold conditions. And we all know that the sympathetic nerve innervates every organ of your body, and there is a basal tone, which is essential for homeostasis. So next, I wanted to ask whether this basal sympathetic tone is also important for stem cell function. To answer this question, I use several models. In all of them, I try to devoid hair follicle stem cells from sympathetic signaling. So first, we ablated the sympathetic nerve, both genetically and chemically. In addition, we also knocked out specifically the adrenergic receptor from the hair follicle stem cells. And in all of these models, we saw exactly the same phenotype. Hair follicle stem cells were more quiescent, and there was a significant delay in their activation. So if you look here early in the control, you can see that there is colocalization between EDU and hair follicle stem cells, which means that hair follicle stem cells were activated and starting to proliferate, while there is no such colocalization when there is no adrenergic signaling. As I said, hair follicle stem cells are a major cellular force that drive hair growth. Hence, in the control, you can see very long, elongated hair follicles, while when there is no adrenergic signaling, the hair follicle remains very, very short. From all these sets of experiments, we can make two major conclusions. First of all, basal sympathetic tone is important for hair follicle stem cell reaction and how they control hair follicle stem cells. The second conclusion is this interaction is a direct interaction, since when we knock out specifically the adrenergic receptor from the hair follicle stem cells, we got exactly the same phenotype. Next, I wanted to understand how this interaction is being integrated into the molecular machinery of hair follicle stem cells. For that, we perform RNA sequencing and compare control hair follicle stem cells to the transcriptome of hair follicle stem cells in which we specifically knock out adrenergic receptor. When there is no adrenergic signaling, hair follicle stem cells have a specific reduction in proliferation machinery. But in addition, there were additional changes such as changes in oxidative phosphorylation and mitochondria function, all of which are very specific attributes of a more deep quiescent state of stem cells. In addition, we saw specific elevation of quiescent markers such as FOXP1 and FGF18, whose overexpression recapitulates the phenotype when we ablated the sympathetic nerve. Next, I wanted to further understand the cellular interaction between the hair follicle stem cells and the sympathetic nerve. You can see here the sympathetic nerve in green and the hair follicle stem cells in red. And you can see that the sympathetic nerve really wraps around the hair follicle stem cells, creating multiple of contact points. To further analyze this, the nature of these contact points, we use both high-resolution confocal imaging as well as 3D electron microscopy reconstruction. In all of them, we saw that the terminal axons of the sympathetic nerve create a synapse-like structure with the hair follicle stem cells. If you can see here in the confocal imaging, when the terminal axons approaches the hair follicle stem cells, we can see varicose morphology, which is an indicative of an active site. Moreover, this varicosity near the hair follicle stem cells are also positive for presynaptic marker synaptotagmin. Similar things we saw in 3D electron microscopy reconstruction. This is just a reconstruction of only 2 axons, and you can clearly see the varicose morphology. When you look at the section through the varicosity, you can see that the diameter of the axon is very, very large. It's partially unwrapped from the Schwann cells. There is multiple mitochondria. We were also able to see specific neurotransmitter vesicles. But when you look in between varicosities, the diameter of the axon is very small and it's completely wrapped by Schwann cells. All this indicates that the sympathetic nerve not only has a direct communication and regulate hair follicle stem cells directly, but it do so in a very specific anatomical structure which resemble a synapse. Next, we wanted to understand how this innervation is maintained. And it's safe to assume that the target organ is important to maintain its innervation. And when we're thinking about the sympathetic nerve, there are 2 possible target organs, the hair follicle stem cells, but also this muscle that cause goosebumps. When we partially ablated the hair follicle stem cells, there weren't any gross changes in the innervation pattern. So hence we decided to ablate specifically this small smooth muscle, which you can see here in the control in red and the innervation in green. So when we ablated this muscle were gone, but also the innervation to the stem cells disappeared, suggesting that this muscle is essential to maintain the innervation to the stem cells, suggesting that the muscle together with the sympathetic nerve, both of them act as a niche to regulate stem cell behavior. Finally, we want to understand how this niche developed, and we found out that sonic hedgehog, which is secreted from the hair follicle, is mediating the differentiation of this muscle and the subsequent innervation of both the muscle and the stem cells, suggesting that the developing hair follicle is mediating the development of its own niche. So to summarize what I told you today is the sympathetic nerve creates a synapse-like structure with hair follicle stem cells, allowing for direct communication and directly regulate stem cell behavior. In basal sympathetic tone, makes the hair follicle stem cells poised for activation. When there is increase in such tone, such as for example, in cold condition, and when this cold condition persists, this elevation in the tone pushes the hair follicle stem cells directly towards activation, allowing them to adapt to the different environments. For the future, I would like to use both the conceptual advances that we made with this work, as well as the multiple tools that we have developed in order to further study how stem cells interact with the environment and how this interaction is important in both homeostasis and disease. So as I said, many efforts have been done and to identify these very locally secreted cues, but there are many, many open questions. And one of the questions that I'm interested in, I've been interested in for a long time, and now I have the tools to address it, is how mechanical forces can be directly integrated into stem cell function. And I'm also interested in more systemic factors, such as the nerves and its interaction with additional stem cells within the skin, as well as the hormone control of the niche and stem cells. And with that, I would like to acknowledge all the people who helped this work, especially my mentor, Yachi Shu, who've been amazing, Mariam Gonzalez-Salerio, a former PhD student in our lab, who I work closely on this project, multiple collaborators, and of course, all my funding. And thank you for giving me this opportunity to present.